I'm so thrilled to be with all of you today to share about the international collaborative research model that we've built for Castleman disease and to share it with your incredible community. I've had the amazing honor of working with Ruth Ann Langen-Pai over the years on Castleman disease, and I'm just so thrilled that she is a research director for PSC Partners doing such important work. Today, I'm going to be sharing my perspective as a loved one of a patient. This is a picture of my mom and I shortly after she was diagnosed with cancer. I'll be sharing my perspective as a physician. This is a picture of me while I was a medical school student training to become an oncologist in memory of my mom. I'll share the perspective of being a patient. This is me when I was a critically ill patient while I was in the middle of medical school. I'll share my perspective as a researcher, fighting to try to find a solution for my disease. And finally, that of as an advocate, trying to work together with other physicians, researchers, and patients to make progress for Castleman disease. I believe it's these 5 perspectives that we can bring together through PSC Partners and other rare disease efforts to make tremendous progress for our given diseases, but even across diseases. Another way of framing it up is to highlight patients, physicians, advocates, researchers, and loved ones of patients working together with the FDA and pharmaceutical companies. When we can bring these parties together, we can make incredible impact on an individual patient basis, and you'll hear my journey today. We can make an incredible impact within a given disease like PSC. And finally, we can make a major impact across diseases when we share what we learned for Castleman disease with PSC, with other diseases. This is something that has been so critical to the progress we've made for Castleman disease, is sharing with one another. And I hope that that'll come through my talk today and that it will be helpful for all of you as you work to advance research and treatment for PSC. I'm going to start by beginning with my rare disease journey. Then I'll share about the power of patient centricity and collaboration in international research, share a couple of applications beyond Castleman disease, and then close with some lessons learned. As I shared earlier, I was a healthy third-year medical student. I wanted to become an oncologist in memory of my mom, who had passed away while I was a college student. When out of nowhere, I became critically ill. My liver, my kidneys, my bone marrow, my heart, and my lungs all began to shut down for an unknown cause. You can see this photograph of me here. I gained 70 pounds of fluid. I drifted in and out of consciousness, had a retinal hemorrhage that made me blind in my left eye, all without a diagnosis. I was so sick that my dad reached out to anyone and everyone he could to try to find answers. He eventually got the phone number of a doctor at the NIH who he reached out to and shared laboratory work with every day to see what was going on with his son, with me in the ICU at the University of Pennsylvania. And months later, when I was finally discharged from the hospital, I asked my dad, who was that doctor you were calling every day at the NIH? And he said, I don't really know. I think his name's Fucci. You should Google NIH Fucci. And so I did. And I learned that my dad had been calling Dr. Tony Fauci every day while I was in the hospital. And every day he got back to my dad and shared thoughts on what was going on with me. And I shared this story for a couple of reasons. First is that my dad should have known who Dr. Fauci was. He had already won a Presidential Medal of Freedom for his work. But secondly, and most importantly, I shared this story because it highlights the power of the loved one. So many of you all watching this today are the loved one of a patient with PSC. And we know just how far you'll go to help your loved one that has PSC. My dad would have gone to the ends of the universe to find answers for his son. And I know so many of you all do that as well. And we as scientists and physicians, we have to remember the voice of the patient and the voice of the loved one. Fortunately, after weeks and weeks without a diagnosis, I was eventually diagnosed with a disease called idiopathic multicentric Castleman disease. This is an image of my lymph node, which was resected. And fortunately, with the resection, my doctor said, we think we finally figured out your diagnosis. About 5,000 patients are diagnosed each year in the US with Castleman disease. It's a disease where the immune system becomes hyperactivated, attacks and shuts down your vital organs, for in many cases an unknown cause. Unfortunately, my subtype, idiopathic multicentric Castleman disease, about a third of us will die within 5 years of diagnosis, another third will die within 10 years of diagnosis. So it's really a frightening disease to be diagnosed with. I know you all that are in the PSC community can certainly appreciate just how scary it is to get a diagnosis like this. Not going to go into too many details, but I just wanted to highlight that like PSC, there's a lot of work that needed to be done to understand what was going on molecularly and mechanistically with the disease. So we knew for many years that patients have enlarged lymph nodes, have flu-like symptoms, have organ dysfunction, but we really didn't understand why until Kazuo Yoshizaki, this brilliant physician in Japan, discovered that interleukin-6, a key protein in the immune system, was highly elevated, and he thought that maybe it was critical to the disease. But even with this discovery about interleukin-6 playing an important role, there was always a black box around, well, what's causing the IL-6 to be elevated in the first place? What are the signaling pathways, cell types, etiologies that could explain why interleukin-6 is elevated? Is this an autoimmune disease? Is this a cancer? Is there some sort of pathogen that's causing the hyperinflammation in the immune system? And importantly, thanks to Kazu's work, there were discoveries made that led to trying an IL-6 blocker called tocilizumab, and more recently one called cetuximab, that can block interleukin-6. And amazingly, about 1/3 of patients just get better. This is a picture of me with Kazu doing our Castleman flax. And amazingly, if you treat someone with one of these drugs, 1/3 of patients just get better, which is just incredible. But for 2/3 of patients, you can block interleukin-6, and unfortunately, patients don't get better. So patients like that don't respond, so patients like me that do not respond to IL-6 blockade, we get cytotoxic chemotherapy to obliterate the immune system because we don't know what to target, so we just wipe out the whole immune system. And that's what was done for me. IL-6 blockade didn't work, so I got multi-agent chemotherapy, and thankfully, the chemotherapy worked. You can see in this picture that I'm sitting up. I've got a bald head, but my dad's next to me, supporting me along the way. And then this is a picture of my dad and I on New Year's Eve of 2010. We were going on a walk around the hematology oncology unit, and we passed someone who'd been clearly drinking on New Year's Eve. He was visibly drunk in his chair. And on our next lap around the hospital, we saw this drunk guy had fallen on the ground. And so my dad ran over to him, helped him back into his chair. He looked at my dad and I and he said, "Thanks so much. Good luck to you and your wife." I said, "Wife? What is he talking about?" And then I looked down at my belly and I realized he thought that I was my dad's pregnant wife and that we were going on a walk to deliver our child, which was a low point emotionally for both of us. But we really laughed so hard. And something that I think so many of you can appreciate is the power of laughing with the people that you love. So facing my horrible moments with laughter was just as fundamentally a rejection of Castleman's dominion over me as anything else I was doing. It cleared my mind, it stiffened my resolve. It was entirely up to me to determine what was and what wasn't funny. Perhaps most important, humor is social. For me and my family, there was never a better way to reset our collective resolve than laughing together. So I was eventually diagnosed from the hospital now after a total of about 6 months hospitalized. And you can see that my head's bald, my belly is huge. Maybe you can see why someone might have confused me as my father's pregnant wife. And this is a picture of me from when I played college football a few years before. And I always say these are the worst before and after pictures of all time. But if you could flip the order, it could be this great advertisement for Mirror or Peloton. But unfortunately, the pictures are in the wrong order. I did take a picture every week for the next 8 weeks, and you can see my hair starting to grow back. You can see my belly starting to shrink away, and I look a bit less like my father's pregnant wife. And again, pictures from the side, where again, you can see the recovery thanks to that chemotherapy. It was actually a combination of 7 different chemotherapy agents that really obliterated my immune system and saved my life. When I got out of the hospital, I reached out to a girl I'd been dating before I became sick named Caitlin. And I was so thankful to learn that she also had been thinking about me while I was ill, and we started dating again. This is actually the first picture we took together after we started dating. And you can see I'm dressed as Borat in this picture, who's a role model of mine. And then this is probably my favorite picture of the two of us. And what I love about it is that on one shoulder, I'm getting an experimental treatment, cetuximab, that's getting pumped into my port. On my other shoulder, I have Caitlin, who's supporting me during a really difficult time. I think this is such a good representation of my journey, and I think so many of you all's journey. Medical technology on one side, helping us along, but the people we love on our other side, supporting us along the way. So I was able to return to medical school after being on medical leave for about a year. And that's right around the time that Penn launched an orphan disease center. But unfortunately, I relapsed and I nearly died for the 4th time while on this experimental drug. It clearly didn't work for me. And my doctor explained to me that I was out of options. There were no more drugs in development. There were no more promising leads. And so at that stage, I promised my dad, my sisters, and Caitlin I would dedicate the rest of my life however long that may be, to trying to find a treatment for me and for patients like me. I knew that I couldn't do it alone, so I launched a community called the Castleman Disease Collaborative Network, and we decided we would take a unique approach to advance research and treatment for Castleman disease that we called the Collaborative Network approach, an approach that many of you all in the PSC community have been utilizing and pushing forward, which is really a patient-driven approach to research. Now, I shared a quote earlier from my book, Chasing My Cure, and I'll share another one. This is from a really critical moment in my journey. And that's the realization that hope cannot be a passive concept. I couldn't just hope that some researcher somewhere would find a treatment that could be helpful for me and for other patients. I realized that hope is a choice and a force. Hoping for something takes more than casting out a wish to the universe and waiting for it to occur. Hope should inspire action. And when it does inspire action in medicine and science, that hope can become a reality beyond your wildest dreams. So where were we back in 2012? Well, there was about $10,000 a year being invested into translational research for idiopathic multicenter Castleman disease. The NIH had never funded IMCD research. There were no patients in our registry because there was no registry. There were 2 advocacy organizations, but no sort of research plan or infrastructure, no cell lines or animal models, biobanks. The disease is very poorly understood. The cause was unknown. There was no diagnostic criteria, no treatment guidelines, no predictive biomarkers, so blood tests that could predict whether one drug would work or another, no FDA-approved therapies, and the only drug in development, the drug I mentioned earlier, Cytokine, did not work for me. So there were no drugs approved nor drugs in development that were looked promising in my case. Visualized another way from a diagnostic perspective, there was limited disease awareness, no coordinated patient community, no diagnostic criteria. From a treatment perspective, there were no treatment guidelines, no FDA-approved therapies. And from a research perspective, there was no coordinated research community like you all have in PSC Partners. There's no specific ICD-10 code. There was no biobank, no registry. This is infrastructure that you all have built for PSC, which is really advancing the field. So I want to share some examples of the power of patient centricity and collaboration to get from where we were to where we are today. But before I highlight the infrastructure, I want to highlight one important principle that really is critical to why I'm alive today. And that's the realization that there are about 10,000 human diseases, and there are about 2,500 drugs that happen to be approved for about 2,500 of those diseases. This is incredible. Humankind has developed 2,500 drugs that are approved for about 2,500 diseases. And now some diseases have multiple drugs approved for them, and some drugs have multiple diseases that they're approved for. But in total, there are about 7,000 diseases that don't have a single FDA-approved therapy. And so this is how things were for Castleman disease. And I wondered, could one of those 2,500 drugs that are already approved for something potentially be a treatment for Castleman disease, for something else? And I was hopeful that that'd be the case, because I knew that was really the only path forward that really would work in time to save my life. There was no way I had the time or resources to develop a new drug from scratch, but I certainly could be a part of identifying what was going wrong in my case and trying to identify a drug that could maybe be helpful for me, a drug made for some other disease. But in order to do that, we had to take a new approach to research, this what we call the collaborative network approach, which I wanted to share with you all today because I believe that your community is following many of these principles. So to understand what's novel about this approach, I think it's important to start by looking at what's the traditional approach to research. Well, first off, the traditional approach to research is that patients come together, raise money for research, then organizations will put out a request for proposals to the disease field of interest and invite all the best researchers in the world to apply for that funding. And out of that whole field, a fraction will actually apply for grants. And out of that fraction that applies, an even smaller fraction will be awarded one of the grants to do the research that they propose. The study is then executed, and really in the traditional research approach, you kind of start from the beginning again every 6 to 12 months. And now the challenge with this is that only a fraction of the entire disease field is able to contribute their ideas for what research should be done. Whoever applies for funding, those are the people who actually get to provide their ideas for what should be done. And out of all the great ideas that are proposed, only a fraction of those ideas are actually funded. And importantly, this doesn't involve patients in selecting what research should be done. Sometimes patients are part of the panels that pick the best applicants, but they're not part of deciding what kinds of research should we do as a field? So in essence, you hope that the right application is submitted by the right researcher at the right time. We decided to come up with a different approach. We call it the collaborative network approach, where we don't rely on the right researcher applying for the right project at the right time. We figure out what is the right study based on engaging the entire community so that we can find the right researcher to do it right now. And so the first step in our collaborative network approach is to build a community, first virtually through online means. We have something called CDCN Connect. We connect patients, physicians, researchers online, but then also in person through annual meetings that we hold at the American Society of Hematology and also here in Philadelphia for patients. So we connect the disease field, so physicians and researchers with patients virtually and also in person. And importantly, we also look into researchers in related fields, diseases like Castleman disease that either treat or research those related fields. Connecting them all virtually is the first step. The next step is to crowdsource from the entire community, to ask all of our community members, physicians, researchers, and patients, what research questions are important to you? What research studies could be done to address those questions, and what researchers are the best people in the world to answer those questions? Patients are really good at being able to tell us what research questions are important to them, and that should be our northern light. That should guide us towards what we fund and what kind of research we do. So we build a community, we crowdsource, we identify from the community what are the most important research questions that we should be asking, and then we recruit all researchers to propose research ideas to address those particular research questions. And we go out and we proactively recruit people, even people who don't know anything about Castleman disease. And we'll say, our community prioritized improving diagnosis of Castleman disease, and proteomics was the platform recommended. You're the best person in the world for proteomics. We want you to do proteomics for our disease. Even if you've never heard of Castleman disease before, we think you're the right person. We'll give you samples and we'll give you money and we'll give you data. Will you work in our field? And this has been really successful for us. Next, once we have the community built, the right studies to be done, the right researchers to do it, then we raise money for that particular project. We work directly with patients to recruit them and get samples from those patients, get data and participation and engagement from those patients in the study. We perform the study, of course, track its progress. We support the analysis, and we always look for drugs that can maybe be repurposed. Like I mentioned in the slide before, thinking about drugs that are already approved for one thing that could be repurposed in another way. We disseminate that knowledge to our community from the study. Then the community gets better and better at crowdsourcing the next set of studies, the next set of research ideas. And so it's a way to improve knowledge and also to continue to get better and better at crowdsourcing research. So this sort of an approach has really been a game changer for the Castleman disease community. I know you all have embraced it within your community, and I'm always eager to support any way that we can and to hear about the progress you all are making. So once we came up with this game plan, this blueprint for how we were going to advance research, the next thing we did was to turn to our patient community and to hold a webinar to connect with them and say, this is our vision. We want to hear from you. We want you to prioritize research. We want you to contribute your samples and your data so that we can figure this thing out together. So we held a webinar, and I showed this picture of a Castleman. I Google imaged Castleman. This is the first thing that came up. And our community liked it, but felt that maybe we could do a better job. And so we went back to the drawing board and we created a new Castleman that's much more fierce. And you can see that Castleman here in this photo. So we held another webinar a few weeks later and showed it to our community. One of our patients screenshotted the image and then got it tattooed on his shoulder and then posted it on our Facebook page. And I always say, if this is not patient engagement, then I don't know what patient engagement is, but there are literally hundreds, maybe even thousands of Castleman warrior tattoos on patients all over the world. Our community was just waiting to get behind something. And by creating the CDCN, we've been able to give our community that initiative to get behind. These are just some of the many Castleman disease patients that are part of our community from all over the world. It affects individuals of all ages, races, genders, and we are just so thankful to be able to support them with the work that we do, and not just to support, but really engage and empower them to be part of this effort. So once we had the right research plan, the patient community assembled, the next step was to identify a team of volunteers. And so I turned to my classmates in medical school and business school to be a part of our efforts, to be a part of identifying and prioritizing the right research questions so we could really push things forward. And I see my good friend and colleague, Ruth Ann Langen-Pye, who's a part of you all's effort with PSC Partners, photograph in a couple of these pictures. She really was such a critical member of our team as a PhD student in my lab. And I'm just so thankful that she's now part of your community. In this picture, we're doing something called the Castleman Flex. If we were all in person today, I would have you all stand up and do a Castleman Flex. But I think the best thing I can ask for is that maybe some of you all will do some Castleman Flexes and then post it on social media to help to raise awareness about Castlemans and the work that we're doing. Next, we turned to our physician and researcher community, and we identified anyone and everyone who had treated any Castleman disease patients, also anyone who had either studied Castleman's or any related diseases, and we connected them through this CDCN Connect platform, which now has hundreds of physicians and researchers from around the world. Kind of shown another way, our blueprint, in addition to crowdsourcing research questions, it was really let's crowdsource research questions so we can really understand what's happening biologically and then translate that into treatments. And so we have a biobank where we get high quality biospecimens from patients. We have a registry where full medical records are obtained from all of our patients. And then we utilize the biobank specimens and the medical record data to then be able to perform experiments on those samples so we can identify aspects of the disease that we didn't even know to look for. And then we use that as insights to then think about, okay, if we figure out a particular cell type might be important to Castleman's, what happens if we eliminate that cell type with a treatment? How can we modulate these therapeutic targets and what happens? And when we do that, we track how that drug works or doesn't work in our registry. We take the most promising drugs and move them forward to clinical trials. Then we perform correlative analyses on proteomic data, or in this case, proteomic data from the SPEED2 study to get predictive biomarkers of who's likely to benefit versus who's not likely to benefit. We really translate all this information back into our better understanding of the disease, and that can lead to continued cycling through of this process. Don't have time today to go through all of our studies, but you can go to cdc.org/research and you can see the progress we've made since 2012. When back in 2012, there was one study underway. Now we've got well over 20 studies either underway or recently completed. So in the midst of this progress pushing forward our international research agenda through the collaborative network approach, I was able to graduate from medical school. This picture of me and Caitlin on my graduation day. I enrolled in business school because I realized that some of the greatest hurdles for progress were organizational problems. But then unfortunately, I relapsed and I nearly died, this time for the 5th time for my disease. Fortunately, Caitlin and I had gotten engaged to get married just a few months before I relapsed. But unfortunately, I knew that I wouldn't make it to our wedding date unless we were able to identify a treatment that can maybe be helpful to keep my disease in remission. So I went back to the sample that I'd been storing on myself. I ran proteomic analyses on my serum. I looked at the cytokine data that we had in my medical records. And we also performed flow cytometry of my circulating immune cells, and bringing all this information together and then applying pathway analyses against the proteomic data, we're able to identify the particular communication line called the PI3 kinase AKT-mTOR signaling pathway seemed to potentially be activated in my samples. And so we wanted to validate that in the laboratory. Could the mTOR pathway be kind of a central node or something that's an important pathway in my disease? So we then stained my lymph node tissue to see how much mTOR activation there was. So first looking at normal lymph node, and you can see here in this normal lymph node that most of the cells stain negatively for mTOR activation. Most cells are blue, but there are some cells that are brown, meaning that there is mTOR activation occurring, but it's really the minority of cells. And if you look at my lymph node, you're really blown away by the tremendous number of cells that have increased mTOR activation. It's really blazingly positive, confirming that this particular communication line was turned on. This communication line is really important for a number of the biological processes that we see in Castleman disease. So it really would make sense that maybe if this communication line is turned on in the immune system, then maybe that could explain why the immune system is getting out of control. And so we really thought mTOR could be that central aspect. Now, fortunately, there's a drug called sirolimus that's been around for decades. It was initially developed as a kidney transplant rejection drug. No one had ever thought to try it for Castleman disease. But here I was 50 years later with data that made me think that maybe if we could block this particular communication line called mTOR with this drug called sirolimus, maybe we could save my life and prevent a relapse. Now, today marks 100.93 months. And I say 100.93 months because I know that I can't round up. I don't know if I'm going to make it to 101 months in remission, but I also refuse to round down. It's not 100 months. I'm so thankful for every fraction of a week, a month, a day, a year that I've been in remission. And I hope that you all can appreciate, and I think many of you all with PSC can appreciate that mindset of never rounding up, because we don't know what tomorrow will hold, but also never rounding down because we're so thankful for every fraction of life that we have, thanks to incredible scientists and medical technology. Now, during this remission, the New York Times called this "Doctor Cure Thyself," which I think is a bit of an overstatement. I think it should be doctor helping himself a little bit right now and hopefully for a lot longer. But I don't think that that would have fit in the headline. Importantly, during this remission, Caitlin and I made it to our wedding day, May 24th, 2014. We were able to get married. My hair grew back just in time for the wedding photos. And then about 3.5 years ago, we had our first child, Amelia, photographed here with us. And about 6 months ago, we had our second child, baby David. And as I look at these pictures, it really feels like a dream that I'm here and alive this many years later on a drug that no one had ever thought could be useful, thanks to a process, the collaborative network approach, that had never been tried before for biomedical research, that really has been a game changer for the Castleman disease community. Since this time, I launched the Center for Cytokine Storm Treatment and Laboratory at the University of Pennsylvania, and more recently started something called the Corona Project when COVID-19 emerged, to apply what we've learned about hyperinflammation from Castleman disease to COVID-19. Once we figured out that sirolimus was helping me, we started giving it to other patients like Katie, who was one of the first patients to receive it. Patients like Deepa, Clara, Jacob have all received sirolimus and benefited from this drug. And it's just been incredible to see a drug that was not made for Castleman disease, for something else, that's now helping these patients in ways that we never could have imagined. Also, patients like Joey have also benefited from this. Kyla as well. Michael Fajgenbaum has benefited from a different drug, as Kyla has. And so we've applied this same approach to drug repurposing, to find new uses for existing drugs, like a JAK inhibitor for Kyla, a PD-1 inhibitor for Michael for his angiosarcoma. This sort of approach of trying to figure out what's going on in a disease and seeing, is there a drug that's already FDA approved that can maybe be useful. And actually, it was Ruth Ann Langenpei's work that served as the foundation for us trying this JAK inhibitor in Kyla's case. But unfortunately, despite all of our best efforts and following drug repurposing for every patient that we can, patients like Lisa and Elise and Joseph here, they have not been able to benefit from our treatments. And so unfortunately, each of these patients passed away battling their Castleman disease. And they remind us that we have to continue to push forward to find more and more treatments for patients like them. So where are we today in 2022 as we continue to search for more and more solutions for patients like the patients I just mentioned? Well, there's been about $1.8 million invested into translational research for IMCD, which has led to an additional $11.6 million donated. So a total of $12.4 million has gone towards IMCD research. So we've invested 1.8 really efficiently, which has led to external investments from other organizations, which has really just been a game changer for IMCD. One of those external organizations is the NIH, finally giving the first ever IMCD grant for Castleman's. We now have over 1,200 patients in our registry. We've got one unified collaborative network. We've got a unified research agenda. We've got a biobank with animal models and cell lines in development. Many questions still remain, though we have made important progress. We've got diagnostic criteria for unicentric Castleman disease and multicentric Castleman disease. We've got treatment guidelines for both subtypes of Castleman's. We have blood tests that can predict whether one drug is going to work better than another. Siltuximab went on to get FDA approval, so we have one FDA approved drug. And now there are additional drugs in development, both the drug that I'm on, sirolimus, we're trying that in other Castleman's patients through a clinical trial, and also the drug ruxolitinib that I mentioned is helping that patient Kyla. We're now also pursuing JAK inhibition as another approach. So looking at this another way, I showed this slide earlier for where we were in 2012. Well, now in 2022, we've had tremendous international disease awareness. We have coordinated community events. We've got a published diagnostic criteria. We have published treatment guidelines and FDA-approved therapy. We have a coordinated research community, a specific ICD-10 code, a biobank, a registry. Bringing all these things together really creates an infrastructure and an environment that enables progress and breakthroughs for patients like me. I just have a few slides left. I'm going to close by sharing some examples of other communities that have really united to advance progress. So I shared earlier about our natural history, or about our work, and specifically we have a registry called Accelerate, bringing together medical record data also with omic analysis. There are organizations like the Chordoma Foundation that really have emphasized connecting physicians and researchers for continuous scientific discourse throughout the year, not just at the annual meeting, but also having virtual platforms to continue to communicate throughout the year. The Rare is One project, which you all through PSC Partners are a part of, was actually started by an incredible group of leaders at Chan Zuckerberg Initiative with the goal of scaling this approach that we've pioneered for Castleman disease to many more rare diseases. Can we make sure that all rare diseases have a patient-led initiative that's crowdsourcing and generating an international research agenda and really pushing forward the science? There are also efforts like the Count Me In initiative and the Angiosarcoma Project that have pioneered directed patient consent for samples and medical data. We follow this approach. We consent patients all over the world with Castleman disease to get their blood samples, to get their data. And this certainly has been a game changer for many diseases like angiosarcoma, but also systematic tracking of off-label treatments. If drugs are going to be used in your disease area and they're not approved for your disease area, it's critical that you have in place a system to track how well those drugs are working or not working. We have to build upon that information to help future patients. And I mentioned the Corona Project, which I don't have time to go into today, but it's an effort we launched to centralize all data on COVID-19 treatments being used around the world. And we were able to use that to help to prioritize the most promising treatments for large clinical trials. It's really been an honor to be a part of that. I won't spend much time on this, but just to say that drug repurposing is a really promising approach for identifying existing drugs that can be used in new ways. And there's multiple ways to identify and validate potential candidates. But at the end of the day, you got to give the drug to humans, whether it's through off-label use or through clinical trials. And then you have to deeply evaluate whether the drug's actually working or not. And that can lead either to FDA approval or to adoption in clinical practice, even without FDA approval. So I'm just going to close. This is my last slide highlighting some of the lessons from my journey. I shared earlier that I wrote a book about my journey called Chasing My Cure. And it's been so incredible to hear from readers all over the world about what that book has meant to them. I wrote it because I learned so many lessons about life, living, hope, chasing cures from nearly dying 5 times, lessons that I didn't want others to have to go through all that I went through to learn, a playbook truly for anyone coping with an illness themselves or an illness that their loved one is dealing with. And so I was able to share those lessons through this book, and now I'll try to share a handful of those lessons with you all today. So the first is that there's incredible power when patients drive research. And I know that patients are really in the driver's seat within the PSC Partners community, and I hope that that will continue to be the case. Second is that laughter can be so powerful. I hope that no one watching this is ever confused as your father's pregnant wife. But if you are, I hope you'll find some humor in that and all of life's challenges. Third, we have to turn our hope into action. My life changed when I went from being a very hopeful person to realizing that if I'm going to hope for something, I need to act. And I know you all, by being a part of this effort, you're acting, you're giving your blood samples, you're giving your medical data, you're sharing your ideas, you're working with your community. These are the kinds of things we have to do. We cannot just hope and we can't just act. We need to be hopeful and we need to take action. Fourth, solutions can be hiding in plain sight. I mentioned earlier the 2,500 drugs for about 2,500 diseases. Well, the drug that I'm on was sitting at my neighborhood CVS for all the years I was in and out of the hospital. But thanks to this work, we were able to discover that actually there was a drug that already existed that could be useful for Castleman disease. We've also gone on to discover that there are drugs that already exist for COVID that can be repurposed. And we now are working to figure out ways to leverage all of this data, all these samples, to find as many possible uses as we can for as many treatments as we can. And so really, I want to close with my last lesson, and that's that my book is called Chasing My Cure, but it really should have been called Chasing Our Cures, because it really does take all of us working together. And in our case, it's really been an army of people working together. I hope you all will become part of that army, helping to raise awareness about the collaborative network approach, about Chasing My Cure, about the journey that we've gone on. And I'm just so excited. I'm part of your army, cheering you all on, and just excited to see the progress that you all will make following this approach to research. And of course, I just want to close by thanking my incredible team, our collaborators, and everyone who's been a part of this effort to advance research and treatment for Castleman disease and to turn my hope into action. Thank you all so much. And I'm really excited to answer any and all questions that you might have.